Welcome students. My name is Ms. Karen Lenisha Cabral, Assistant Professor in Botany at St. Joseph Vaz College, Kottalim. Today's lecture is for third year Botany students, Semester 5, Paper Code BOC 107, Paper Title Microbiology and Plant Pathology. Title of the unit Applications of Microorganisms, Module name Antibiotics, that is Streptomycin, Module number 29. Outline, Introduction, Types of Antibiotics, Discovery of Streptomycin, Process of Streptomycin fermentation. Learning outcomes, lists the type of antibiotics, recalls discovery of streptomycin, describes the process of streptomycin fermentation. Introduction. Antibiotics is a substance produced by microorganism that inhibits the growth and replication of bacteria. Now, when we talk of antibiotics, there are two types of antibiotics, or we can say they can be categorized into two groups again. We can call them as broad spectrum and narrow spectrum. Now, when is broad spectrum used, or antibiotics which are called as broad spectrum, they can be used against many infections. When we talk of narrow spectrum, they are specific to a particular infection. Now, when we say antibiotics, it is strictly for bacteria and not viral infections. So they are specific for a type of infections, that is only bacteria. And antibiotics, depending upon their mode of action, can be either bactericidal, that is it kills the bacteria, or it can be bacteriostatic, that is it prevents the growth and reproduction of bacteria. These are a few types of antibiotics that are used. In this particular module, we are going to refer to streptomycin, and we'll see how exactly it is produced. Coming to discovery of streptomycin. Now, the antibiotic streptomycin was discovered soon after penicillin. That was when penicillin was introduced in medical field. It was discovered by American biochemist Salman Waxman, Albert Schutz, and Elizabeth Bugge. That was in the year 1943. While penicillin was introduced, or it was discovered in the year 1928. Streptomycin is an aminoglycoside antibiotics. Now, there are here some examples of streptomycin which are listed, that is gentamicin, then you have neomycin, which are used against bacterial infections. Now, streptomycin is bactericidal, which means it kills the bacteria, and it is useful in the treatment of infection caused by gram-positive bacteria, especially that are resistant to this particular antibiotic, that is penicillin. Now, how does it act on a particular infection? First of all, it acts by binding to the 30th subunit of ribosomes, and it disrupts the initiation and elongation step in protein synthesis, that is we are referring to translation. It thus stops bacterial growth and by inhibiting protein synthesis. It was the first antibiotic that was effective in treatment of tuberculosis caused by Mycobacterium tuberculosis. Streptomycin is produced by the soil actinomycetes, that is the organism that is used over here is Streptomyces griseus. Now here that is an image that is being shown, which is taken from a scanning electron microscope. Coming to the structure of streptomycin, it consists of three components that are linked glycosidically. That is, the first part is streptotidin, secondly streptose, and the third component is streptosamine. The structure of the same is in this slide. Coming to the process of streptomycin fermentation. Now, industrially, streptomycin is produced by submerged culture method, and it has four important steps that includes inoculum production. The second step is preparation of medium. The third step involves fermentation, and the fourth step involves harvest and recovery. Now let's look at the first step. In the first step, we have the stock culture, that is the stock culture of spores of Streptomyces griseus, which is subcultured. So, in order to get now what we are doing is we are subculturing because we need to get sporulation. So it is subcultured for sporulation, and then this sporulated the spores are then inoculated in flask or inoculum tank so that the mycelium grows, and then this mycelial growth is introduced in the production tank on a large scale. In the second step is preparation of the medium. Production medium contains a carbon source, and the best source that is known is glucose. There are other carbon sources which are also used. Maltose can be used. And it requires a nitrogen source. Now, when we talk of nitrogen source, we refer to peptones, soya extract, meat extract. Magnesium, calcium, potassium, boron, and molybdenum may also be used as mineral source, that is in the medium. Phenyl acetic acid and L-naphthalene acetic acid are used to stimulate the growth, that is it is introduced into the medium, and proline helps in high streptomycin production. Coming to the third step, that is fermentation. Now here again fermentation is highly aerobic, that it requires oxygen, and it lasts for approximately 5 to 7 days. Now first of all, the liquid medium, the sterilized liquid medium is fed to the production fermenter, and after that the inoculum is introduced. That is the spores, the mycelial growth is introduced, approximately around 4 to 5% of it, and the optimum fermentation temperature is maintained to between 25 to 30 degrees Celsius, and the pH is maintained to between 7 to 8. Now, when we talk of fermentation, it needs to go through three major phases. So, the first phase, the second phase, and the third phase. So, in the first phase, it takes about 24 to 48 hours, wherein rapid growth and formation of abundant mycelium occurs, and the pH rises to 8. And in this phase, glucose is utilized. In the second phase, it lasts for 2 days. Streptomycin production takes place rapidly without increase in the growth of mycelium in this phase. Now here what happens, the pH decreases to 7.6. From 8 it is dropped to 7.6. So glucose and oxygen are required in large quantity. And in the third phase, the cell undergo lysis, releasing ammonia and increasing the pH to 8. Oxygen requirement also decreases, and the content of the medium including glucose gets depleted. So in this stage, streptomycin production is ceased. Coming to the fourth step, that is harvest and recovery, mycelium is separated from the broth by filtration. Now this broth is acidified, filtered, and then neutralized. The broth passes through a column containing cation exchange resin, and this column is then washed with water. The antibiotics is then eluted with hydrochloric acid or phosphoric acid. An antibiotic is concentrated at about 60 degrees Celsius. Then the streptomycin is dissolved in methanol and filtered. Acetone is added to filtrate to precipitate the antibiotics. This precipitate is then washed with acetone and vacuum dried. The streptomycin is extracted in pure form as calcium chloride complex. Coming to the summary, streptomycin is effective in treatment of tuberculosis. Now, the organism that is used is Streptomyces griseus, that is, it is used for commercial streptomycin production, And the fermentation steps involve inoculum production, preparation of medium, fermentation, harvest, and recovery. These are a few references. Thank you.